Okay, Science 8, new unit, cells and systems. So let's do our video notes. Okay, so what do living things have in common are some of the questions we talked about. How are cells organized to carry out different functions, and how do systems work together to keep organisms healthy? That's what we're going to look at in this unit. And so let's start with topic 1. So topic 1 deals with living organisms. Life functions. In order for something to be called a living organism, it must be able to perform all of the following. So GRREW, grew, is an easy way to remember these. Growth is all, so this is what all living things need to do to be considered to be alive. They all have to be able to grow. They all have to be able to respond and adapt to their environment. So for example, birds migrate south for the winter. They all need to be able to reproduce. They all have to somehow receive energy. So plants get energy for the sun. Other animals will eat food in order to get their energy. And they all need to be able to produce wastes. So we went through the whole argument whether cars were alive or not. And in the end, uh, cars aren't made of cells, so they're not considered to be living. And there's the acronym that I taught you to help remember all 5 of them, and it really helps for me as well. And those 5 life functions that we looked at on the previous page are common to all living things. We did a Venn diagram for this, and so we can see this is what a candle and human have in common. They do respond to environment. There is energy being used, and they produce waste. However, humans are able to grow and reproduce, candles cannot. And we looked at the levels of organizing. The basic unit of life is called a cell. We have about 70 trillion cells in human cells. Some organisms are unicellular, like amoeba, they're made of only one cell. And multicellular organisms, organisms that have more than one cell, must specialize and organize their cells to perform life functions. And we looked at the level of organization from cells that all work together to do the same job, get organized into tissues, and the tissues all work together, they become organized into organs, and then the organs are a part of an organ system. And we looked at some examples of that. And here is another example. The circulatory system has specialized muscle cells that form heart tissue, and heart tissue is made from muscle cells that help your heartbeat. They organize together to become organs, like in this case, the actual heart. And then the heart is part of a circulatory system that involves the heart, arteries, veins, capillaries, and that is the circulatory system. And a cell is the smallest unit of life that can perform the functions of life. The tissue is a group of cells having the same structure and function. Organ is a group of different tissues that work together to perform a specific function. And then a system is a collection of organs that work together to keep an organism alive. And I just listed a whole bunch of different systems like circulatory, nervous, respiratory, excretory, digestive, skeletal, and musculatory systems. And then here they're showing a plant's shoot system. So they have specialized cells that have thick walls and they tissue strong. So cells organize to become tissue. The tissue provides support for the stem. The stem is the organ that keeps the plant upright. And the main function of the shoot system is to make food for the plant. And then we did the assignment. And then we did this assignment. And then topic 2 deals with the microscope and cells. Okay, so when things are too small to be seen, we need to magnify them. And so that was, one of the impetus for the microscope to be invented. I'm not going to get you to memorize names for your quiz, but it's always good to know what scientists did what. So Anton von Leeuwenhoek made the first microscope. It had 300 times magnification. He ground up lens and put them together. He studied blood samples, pond water, and matter scraped from teeth. And he observed single-celled organisms. He called them animalcules. And we did a lab where we looked at some animalcules. Robert Hooke's the next person to come along. He created an instrument that resembles today's microscopes. He looked at bark from oak trees. He saw tiny box-like structures. He called them cellulae, which means little rooms in Latin. And that present-day word is called the cell. Schleiden and Schwann used the microscope over the next century and observed many cells. They created the cell theory, the idea that everything is made up of cells, and there is 3 things that cell theory says. All living things are composed of 1 or more cells. Cells are the basic unit of structure and function in all living things, and living things can only come from other living things. And then we have the compound light microscope that have 2 lenses, and basically you multiply them together. So if you have 10 times magnification at the eyepiece and 40 times magnification at the objective lens, 10 times 40 is 400. We also have electron microscopes that use beam of electrons instead of light, so they can see in much more detail, invented by 2 Canadians, James Hillier and Albert Priebus from Edmonton. And then early electron microscopes could magnify up to 7,000 times, but today's electron microscopes can magnify up to 2 million times. And when we encounter things smaller than we're able to measure using millimeters, we use a measurement called micrometers. Okay, and so 1 millimeter equals 1,000 micrometers. And so you can see as you change from millimeters to micrometers, you multiply by 1,000. And so we saw the field of view for a low power is 4 millimeters, so it would be 4,000 micrometers, and for a medium powered lens, it's about 1.5 millimeters, so you can see about 1,500 micrometers. And we did some calculations with this, and we can go the other way around as well. And then we did that worksheet. Well, we did this for marks, easy marks. And then we look at the parts of the microscope. And so there's the eyepiece, and there's the tube, and there's the coarse adjustment knob that's only used under low power. There's a fine adjustment that's used under medium and high. There's the arm that you use to carry that microscope with. There's a revolving nose piece. So instead of changing each lens, it's on a revolving nose piece. You just turn it to get to the right lens. There's the objectives, low, medium, and high. There's the stage where you put the microscope, the slides, and there's the slide, and there's some of them have clips to hold them in place. There is a condenser lens in here that we never really adjust. There is a diaphragm knob that you can change the amount of light going through. And then that is your light source. You are responsible for knowing the parts of the microscope. And there it is. The eyepiece is the part you look through, usually magnifies by 10 times. The tube holds the eyepiece and lens at the proper distance, keeps the magnification correct. The coarse adjustment knob moves the stage up or down, or the tube up or down, to bring the object into focus. You only use coarse adjustment with low power. There's a fine adjustment knob used with medium and high power to bring the object into focus. The arm connects the base and the tube. You use this for carrying the microscope. The revolving nose piece is a rotating disc that holds 2 or more objective lenses, usually 3. You turn it to change the lens, and they click into place. So when you're using a microscope, as I taught you, it's important to make sure you feel that slight click. The objective lens magnify the object. They have different powers. Usually the ones we have 4, 10, and 40, but there are more powerful microscopes. So this one with 100 and then times, and then your eyepiece is usually 10 times, gives you a magnification of 1,000. But for junior high, we use 4 times 10, 40, 100, and 400. And the magnification is written on the sides of each of the lenses. We have the stage that supports the microscope slide. The clips hold the slide in place, and there's a hole in the stage allowing light to go from the source to pass through the slide. There's a condenser lens that directs light to the object being viewed. The diaphragm controls the amount of light reaching the object being viewed, and then the light source shines light through the object and makes it easier to see the details. We looked at how to make a wet mount slide. So you put your slide, you put your drop of water, you put whatever it is you're looking at. In this case, we looked at the letter E, and then you put a cover slip on top of them. Really easy to make the wet mount slide. And then there were some pictures that we talked about. And then field of view. So field of view is, when you look to the microscope, how much you can actually see. With low power lens, you have a larger field of view, but the magnification isn't as high. With a higher powered lens, you have a smaller field of view. That is, you can't see as much as the whole thing, but you can see it in much greater detail because there's more magnification. So there's a letter E under low power. Under medium power, you can see the field of view is smaller, but you can see it in more detail. When you get to high power, the field of view is even smaller still, but the magnification is higher, so you can see more detail. Calculating field of view, we're not going to do this part. So there's a formula just to show you what it is, and we did not go through that. And then we know how to use the microscope. We know the parts of it. We did labs with it. That is the end of outcome number 1. And then we'll do a video for outcome number 2 later on.